Okay, so now we can start. So number seems quite okay. So today we are going to have 3 sessions. All of you have seen hop. Okay. So we are publish these 3 sessions because this course is going to be fast track for you people. So that's why we are having some additional session even on the weekends. Okay, so continuing with what we were talking in our previous lectures, we were talking about the DNA fingerprinting, and some of you introduced DNA and genetic material. And then I give you some bit introduction of about what DNA fingerprinting is, what is the history of DNA fingerprinting, and how it came into the picture. And so we started with the Sherlock Holmes thing, and real Sherlock Holmes is your DNA fingerprinting. And by using it, if you have that thing, if you have a DNA sample, then you can find a criminal, and you can save innocent people, and you can really prove who is a real criminal, and you can give justice to victim. Okay, so when you are talking about the DNA fingerprinting, so as I mentioned earlier, DNA fingerprinting it doesn't means the DNA sequencing. Here we generally refer the sequence, but it is not a complete genome sequencing, or it is not a genome sequencing at all. It means you are not talking about the whole genome of an individual. You are talking about just a certain thing. So then a question came, then what is the basis of this DNA fingerprinting? So the basis of DNA fingerprinting, as we are talking DNA, the name is DNA, but DNA, when you're talking about, as you all know, that the human genome, whole human genome is known. So when we are saying this human genome, it means genome of a human. But whenever you are referring a human genome, it is not a genome of all humans. When you are referring human genome is a reference genome. Reference genome means a genome of some few individuals were taken, and that was sequenced, and that was taken as a reference. And all individuals, as we all know, that we all are different, so we are going to have a different genome in us, so that differences in our genetic sequence are going to be there. But how much the difference is going to be? If you just see, if you compare the genome of all individuals, you will find that 99.9% of human DNA sequence are same. Like simply, if you see whatever we are in this group, like you are 22 students and I am 123, so in our total group 23, if you compare our genome, you will find that it is 99.9% identical. If this much identicality is there, then we consider that it's same, but it is not. The difference that you are saying at 0.01, that difference is a huge difference, and that difference is going to make you what we are, how we are very much different. So if I ask you a question, simple thing, like you are now in advance in your course, so can you tell me what is the size of your genome? So just I will like give a simple calculation. Can anybody tell me the size of your genome? What is the size of human genome? Yes, Manaswini?
Sir, is it 3 billion base pairs?
It is 3 billion base pairs. Okay, so is it 3 billion or 6 billion?
6.4 billion.
It is 6 billion. Okay, so whenever you're referring, we just refer only one set of the chromosome. That's why it is 3.2 billion. But actually, you are having two sets of chromosomes. Thing I want to ask you, I'm connected? Am I connected?
Yes, sir.
Am I audible to all?
Yes, sir.
Yes, sir,
Okay.
now you're audible.
So now I would like to ask you, when you're saying this decimal 0.01% difference is there, so how much is the decimal 0.01% of your 6.4 billion genome? What the actual size is, what the actual difference is going to be? So please tell me decimal 0.01% of your genome, how much base pair it is going to be? Okay, so I'm giving you just a 5 minute for your calculation. So you take your pen, pencil and calculate and just get me after 5 minutes. Okay, so this is simple thing, just simple calculation. You not to do any big thing. Calculate it and come back. I will continue only after you will give me that number. So students, so what's your calculation? Anyone of read calculation? और ये सारे सो रहे हैं वीकेंड पे शनिवार को मजे ले रहे हो?
Sir, it's 640 lakh.
640 lakhs. Okay, yeah, that's might be right number. Okay, so you can see when we are saying it is decimal 0.1%, but still that decimal 0.1% in so large human genome, that is in the billion base pairs, even the difference is even in the lakhs of the base pairs. Okay, so simply lakhs are as how many lakhs you mentioned?
640.
640 lakhs means it is simply in the crores. Okay, so 6 crore it is going to be. So it is quite a large one. So this difference that we're saying it is very small, but it is in principle it is coming out a very large difference. So we are playing, your DNA fingerprinting is playing on this difference. Okay, so enough difference that is possible to distinguish, and this difference is enough to distinguish individual from one and another. Only the condition is there where they are the monozygotic twins, or what we are calling the identical twins, because there is no difference. The genome of two identical twins is 100% same. There is no difference. Okay, so you can't distinguish them, but any other individual like you and me, our both genome is going to differ with this decimal 1%, decimal 0.1%. So we can differentiate. So now the question came where this difference is, in what form this difference is. So most of the differences are there in your intronic regions. So two things you're having, you all know that there are the exons and introns are also there. So in these exons and introns that we are talking about, so the difference is there in the form of repeats. Repeats means a repeated sequence, like if you have a sequence ATGC. So if the sequence ATGC is continuously repeating ATGC, 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 that we are calling it the repeats, or these we are also called the VNTRs or STRs. In detail we will discuss just a moment. Okay. So to that we are calling that VNTRs and STRs. So in details what they are, to what if the sequence, what types the sequence is there, to that we can call VNTRs and WTR. Then one more thing came that these VNTRs and NTRs also one more thing, when you come in the thing that when you want to connect. So dissimilarity we are saying, but on the basis of similarity also we have to talk about. So then only we can connect. So some more things are there, that is a Y chromosome. That is thing with the Y chromosome is that it is going to be there only in the males. No female have the Y chromosomes. And it is a paternal hierarchy and paternal heredity. So from the male it is going to, from the father it is going to the son, and from the son it is going to the his son, so on. So then what's with the females? So females also have a things that we call the maternal hierarchy or maternal heredity, that is the mitochondria. Mitochondria always go from the female to male or female both. So whatever the offsprings are there, if you want to check the maternal hierarchy or the maternal heredity, that you can get from the mitochondrial genome. As if you remember, I think Yukta was giving a talk about the mitochondrial genome. She mentioned that this is a genome that is inherited through the maternal only. So only maternal parents are transferring this genome. So in this way, you can connect whether you want to connect with the paternal or the maternal thing. So this is a way. These are the basis of your DNA fingerprinting. So whatever in the DNA fingerprinting we have to talk about, we are going to talk about on the basis of these VNTRs. That is going to be repeated sequences, or the Y chromosomes or the mitochromosomes, because these are the one thing which are going to create differences, and these differences we are going to mark whether they are different or whether they are going to be same. Okay, so this is then come out. The next thing is how to get the DNA fingerprint, how you can generate your DNA fingerprint. So the DNA fingerprinting, first thing is you need a sample, and sample must contain DNA. So the source of DNA you need. So if you do not have the source of DNA, then you can't get a DNA fingerprinting or a DNA profile. Okay, so then what can be served as a source of DNA. So DNA things means the living remains are the source of DNA, like your blood, your hair, your teeth, your bones, your saliva, your semen, your vaginal excreta, your rectal excreta. So all these can be served as a source of DNA. So all these are going to be a source of DNA, and you can collect these sample there to get DNA. Okay, so for that purpose, if you remember when we are talking about the Alex Jeffries experiment, or Alex Jeffries, how he find a criminal, he take a blood sample. So blood was taken out, so they isolate the DNA from the blood. But every time you can't get a DNA from the blood. Like if a crime scene, like whether it is a rape case. So in the rape case, if you remember, not just a rape case, you can say, or any sexual abuse, we can say. I think some of you have heard about the Monica Lewinsky case. Have you heard about the Monica Lewinsky? Have you heard about the Monica Lewinsky?
No, sir.
No, sir. No, sir.
Have you heard about
Yes,
Bill
sir.
Clinton? Okay.
Yes, sir. Yes, sir. Yes, sir.
विवेक ने तो सुना ही होगा, विवेक तो अमेरिकन है भाई. Okay, so have you heard about Bill Clinton?
Yes, sir.
बाकी ने सुना है?
Yes, sir.
So Bill, who was a Bill Clinton?
Sir, he was a former president of the United States
He
who
was
got caught in an affair with Monica Lewinsky.
terms, and impeachment case was also procedure was also made on Bill Clinton. The reason was that he have sexual relation with his worker, his colleague, his employee. The name was Monica Lewinsky. So then she claimed that she had a relation with the Bill Clinton, and Bill Clinton was a president at that time. So then how it was being proven that she had some sexual relation with Bill Clinton? So that she presented clothes. She presented her clothes. On her clothes, the semen sample were there, or semen spots were there. So that semen spots present on the clothes were taken, and DNA was extracted out from that inner garments of Monica Lewinsky, and from that DNA sample, DNA fingerprint profile was generated, and that DNA profile was checked or matched with the Bill Clinton DNA profile, and it was found that both are the same. So if the inner garments of a lady having semen spots, so then that proves that two individuals are having sexual relation. So by this, it was being proven that the Monica Lewinsky and your president of ex-president of United States, they are having the sexual relation. So these are the things that so simple things that from where you can get the what can serve you as a specimen for your DNA sample. So how to collect sample for the your DNA fingerprinting. So these are the things like that you can say blood, your saliva, semen, vaginal lubrication, and hairs. So wherever you can feel that you can get your DNA. So these or in case of like many cases are there like nowadays sometime it happened that dispute was there whether the flesh or the meat that you are having, whether that meat is of a cow or meat of a pig. So like you know that there is a religious sentiments are attached with both pigs and your cow. Muslims don't like to eat pig, whereas the Hindu don't like to eat cow or beef. So that's why to check whether the meat or the flesh that is present there it is of a cow or if it is of a goat or it is of a buffalo or it is of a uh, any other animal or a pig. So for that also their flesh can serve you as a sample where you can generate your DNA profile. And recently, as I was mentioning, in case of your CDS Vipin Rawat, so there the dead remains that can also serve you as a specimen for DNA extraction. And one more thing, not just these, even your fossils. Fossils, they are going to be there, and the fossil unit at even the fossils of your Harappa and Mohenjo-daro period, and fossils of you can say even this Egyptian civilization period. In your mummies, mummifications is there or simple they are dead remains. So from there you know that everything is being decomposed, but bone remains. And within the bone you say you know that the bone marrow is there. So these bones they also can even we know that the bones are mainly contain your calcium, but within the bone your bone marrow will remain protected. So that can serve you as a source of DNA also, that can serve you as a source of sample. So these are the few things from where you can collect your DNA. So now simple thing is that how to collect your DNA, what specification, what precaution you should take care while you are collecting your DNA. Because thing is that when you are collecting your DNA, so like I think many of you have seen CID. CID देखते हो? CID देखते हो?
Yes, sir,
सर
Dr.
बहुत
Sahu.
पहले देखते थे.
Yes, sir.
क्या नाम है
सर
डॉक्टर का?
देखते
डॉक्टर का क्या नाम है?
Dr. Saluke. Mrs. Saluke.
डॉक्टर सारुके
Dr. Salunke. Yes, Dr. Salunke is the right name that you mentioned. So Dr. Salunke was a forensic scientist. He's a DNA profiler, or he's creating all these things. He's checking whether it's matching or not. So when they were collecting specimen, when they were collecting your evidences, or when they were collecting sabut or proofs, so how they were collecting it? They were wearing gloves, they were wearing shoe covers, they were wearing mask, they were wearing your lab coats, they are covering their head also. So thing is that when you are collecting a specimen that you need for DNA fingerprinting, then you cannot take any chance of contamination. So what can serve the contaminant? What can be the contaminant? Anything can come from the surrounding can add to the contaminant. Like even the sample collector who is collecting the sample, if he is not wearing head cover, then his hairs, because generally we know that at age then the hair fall is there, and not at in the older age, every age there are going to be certain hair fall. एंड लंबे बाल हों किसी के आदिश जैसे और बाद बालों में हाथ फेरने की उसकी आदत है, तो then chances are there that their hairs may fall there. And one more thing, so many people have habit of spitting, okay, and somebody have spit it there, okay, so you can't take fraction or sample collection for granted. So while you're collecting your sample you need to take bit extra precaution. Extra precaution in a way that you can't contaminate your specimen, because if you contaminate your specimen, that can add your DNA to it, and you may, if you found that if the collector's DNA is there, then that profile is not going to be acceptable, or that may prove that collector may also be a criminal. So that's why you need to be a the first important thing in while generating your DNA profile or taking sample for DNA fingerprinting is collecting your DNA sample. Okay, so all the precautions, all what I mentioned, wear gloves, wear mask, wear shoe cover, wear head cover, and collect sample very precisely. Because here even the collection, what you have to collect, here your aim is to collect the specimen, not the surrounding. So so in that case you need to mark which area you have to collect your sample, you have to take photographs, everything you need to do, because each and every time it is not possible to take blood, because if blood is not there, or this because the criminal is not going to come to you and ask to take my blood sample. So whatever the sample, whatever the specimen is available there, you need to take out your DNA from that itself. Okay, so this is the thing. So making DNA fingerprinting, so first thing is you need to collect your sample, and then one another important thing after the collecting sample, the most important one is the transport. So transport means what? Transport means from the site of collection to the Salonke's laboratory. So when the specimen being taken from the site of crime, or the site of from where you're taking DNA sample, then you need to move to the laboratory. So during that process, you cannot allow the degradation of your DNA. Whatever degradation can happen at the site of collection, that is, we can't avoid that one. But from the site, after the collection to the final step of reaching to your laboratory, so that's why you need to collect your specimen in some collection media, or keep it at a lower temperature. So like simply if any one of you have given sample for your COVID testing, what you're doing, you're taking specimen, you're taking swab, throat swab, nasal swab, and then you are putting into the transport media that you call the VTM, viral transport medium. So here also when you're taking specimen, you have to transfer it to a medium which protect your DNA from the degradation. Okay, and also you need to keep at a lower temperature, because if the temperature is going to be higher, that can speed up the process of the degradation of your DNA. Okay, so just what this slide is showing, DNA fingerprinting is a laboratory process that requires DNA isolation. So before DNA isolation, an important task is the DNA, your sample collection or specimen collection. So how to collect your specimen, that we mentioned all of these are the such simple thing. Using your equipments, they must be clean, and you need to identify the spot from where you have to collect, what you have to collect. So here we can't neglect anything, so you have to collect everything. Okay, so then when you reach to the lab, because we are the laboratory personnel, when it is come to us, we can play a role in our laboratory. Means Dr. Salonke is not going to the collection site. Dr. Salonke is remaining in his laboratory. But who is though? So then when sample came to the laboratory, then the job of a DNA fingerprinter or a DNA analyzer is going to come. Like our role is going to be coming to the lab. So our role is the first, that's the DNA isolation, or the collection of DNA. So what techniques we should use, as these samples are very, very special. You can't use the techniques that we are using generally in our laboratory, because in our laboratory we do not have a scarcity of sample. We have ample amount of sample. But in case of when you're talking about the DNA printing in terms of forensic, then there is a very low amount of specimen is there. From that very low amount of DNA, you need to use what technique we are going to use. We'll discuss. That is the part of your module number 2. Just a moment, something happened. Okay, so then next thing is generating the profile. So when you are generating your profile, how to generate, what tools you should use to generate the profile, so that what different tools we are going to use, that will talk in module number 3. So that is going to talk about the tools that you need for generating a DNA profile, to whom you are going to call it DNA profile, what means by DNA profile, that will talk in module number 3. Then next thing is going to be analysis. So in the analysis, we can't like, as a DNA is there, so DNA, as we all know that electrophoresis is an important tool in understanding, studying the DNA. So electrophoresis we are going to use here also. But electrophoresis, when we are doing electrophoresis within the gel. So gel is not very easy to handle, or it's not long lasting, and it's very much chance to breakage of your gel. So you can't store your gel for a longer time. So for that we need to transfer it. So that's why transfer is going to be there, that we know there are southern blotting, northern blotting, western blotting. So all these type of things we are going to do. That's the transfer of DNA to nylon. So this is also going to talk about, I think, module number 3 or 4. Then coming to the probing. So probing is also part of your blotting. Southern blotting means you need to identify the bands, you need to identify the DNA, you need to identify the sequence, whether the sequence is present or absent. So then after doing all these things, whatever you are going to get, to that we are going to call as the DNA fingerprint or the DNA profile. Okay, so this DNA profile is going to be analyzed by you as a scientist, and we are going to deal with it. And on that basis, we are going to compare, and this profile is going to tell you the similarities and differences. If differences are there, differences, as I mentioned, it's however it is decimal 1%, decimal 0.1%, but it is quite huge difference, and that difference we will analyze. Okay, so this is when it is done, then you can say that the specimen that you are having is matching or not matching. If it is matching, then you can claim that the person was present at the site of crime, and if it is missing, you can say that the person is absent. And one more thing is that when we are saying this DNA fingerprinting doesn't always mean the forensic science or a crime. This DNA fingerprinting also you can utilize in analysis of or comparison of different individuals, also different characteristics also. Like simply, if I mentioned you that you have a plant, like how DNA fingerprint can be utilized in plants. So like if you, or how it can be utilized in animals also. So like in case of animal, you know that a gir cow is an Indian cow, and the milk it produces, that is going to be around 30 liters of the milk it produces in Indian condition, Indian cow, and that you know that it's A2 that A2 milk, or what is you are showing A1 and A2, yeah, it is A2 milk it is producing. So which genes are going to be there? If you can, like your Jersey cow, it is also going to produce a high amount of milk, but it is going to be A1 type. So the genes are responsible for this A1 and A2. That also you can find on the basis of on the utilizing this DNA fingerprinting. You can generate the profile, as I mentioned, A1 and A2. So difference is there. So with gene you can map the difference. Like this is the one, this is the characteristic I want to find, and this characteristic is different on the basis of this gene. Similarly, you can utilize this also for finding the, like simple, this problem is there in case of, if have you heard about the papaya? So do you know papaya? Issue with the papaya? What kind of papaya is a plant? Papaya is a bisexual. So it means male plant is different, female plant is different, and some plants are there on which bear the both male and female organs. Okay, so in human we always like boy, 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 boy. But other than human, we like girls, 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 girls. Similarly, in case of cow, we like cow should give birth to female cow. Even in case of plants also of such type, we need only a female plant. Because the thing with the female cow, because female cow is going to give you the milk, male cow doesn't give you, male bull doesn't give you. And in case of plants also, female plant is going to give you fruits, male plant doesn't give you anything. one male plant is able to spread so much pollens, then they can pollinate a large number of female plants. So we don't need male plants. But on the basis of seed, you can differentiate which seed is going to give to you a male plant and which seed is going to give you female plant. Now, how to found if you try to use seed as a source of DNA, then your seed is gone. You can't utilize that seed. You can't use that seed. So we can't check at the seed level, but we can check at the seedling level. Because once the plant is grown, because plant growing take a longer time, when it bear the sex organs, then it took time whether to be getting flower, what type of flower is going to come to it. It take around 2-3 years. You can't wait for that much time. You can't spare so much land for the male plants. So for that, the seedling level itself, you can check using DNA fingerprinting whether whether the plant is a male or plant is a female. Just cut all the seedlings and check, generate their DNA profile and check whether it is a male or it's a female, and exclude all the male, uproot all the males and keep the females. And similarly, do process again. So within a month time, you can tell that your garden is going to have how many numbers of females and how many numbers of your males, because male requirement is very little. Okay, these are the simple thing that I mentioned you. This is just a introduction by your of your module number 1, and with this we end our module number 1. And DNA and RNA, everything, and mitochondria, etc. you all have mentioned. But I think in our next session I will give just a quick run on the DNA structures and something that if you feel that things are missing, I will talk about that also. Okay, so now we are end of our session. So please turn on your camera while call your name. So those students who are joining late, I'm going to mark them absent because okay, for this session I will excuse them, but for the next session, if anyone of you joined late, I will mark you absent. Okay, I will download your Excel file and check your login time. And on that basis, I will mark you absent. Okay, so please, when I call your name, please turn on your camera. So first is Adish.
Present, sir.
Ayushi. Demetrius.
Present, sir.
Devanshi.
Present, sir.
Ishita. Janvi.
Present, sir.
Kshitij.
Present, sir.
Manaswini.
Present, sir.
Namrita.
Present, sir.
क्षितिज तुम्हारा कैमरा अभी तक ऑन नहीं हुआ है. नंदिनी.
Sir.
नेहल.
Present, sir.
साहिबा.
Present, sir. Present, sir.
शक्ति.
Present, sir.
शिवानी. तन्वी.
Present, sir.
त्रिदेव. विवेक.
Present, sir.
गुप्ता. युवराज.
Present, sir.
Okay, so thank you all. So Atoiba is
Present,
absent.
sir. Present, sir.
Okay, so except Atoiba, all are present. So thank you all. So we'll join back at 2:15 for our next session. Till then, stay safe, stay happy.